I'm excited to introduce and to welcome our next speaker, Dr. Martin Jonikas. Dr. Jonikas got his bachelor's from MIT, actually in aerospace engineering, before transitioning fields into biochemistry and molecular biology. He received his doctorate from UCSF in Jonathan Weissman and Peter Walter's lab. He was next appointed as a staff associate at Carnegie Plant Biology over at the Stanford campus, before moving on to Princeton, where he is now associate professor. Dr. Jonikas's research used Chlamy as a model organism to study a unique phase-separated organelle called the pyrenoid, which is involved in carbon fixing for photosynthetic organisms. His exciting work has received numerous awards, highlighted by his recent appointment as an HHMI investigator. It is with great pleasure to introduce a member of the Carnegie family back. Please welcome Dr. Martin Jonikas.
Okay, thanks very much, Zach, for your kind introduction. I'm really grateful for the opportunity to participate, and I'm sorry I can't be there in person today. As Zach mentioned, I started my lab at the Carnegie Department of Plant Biology, and I'm very grateful for the nurturing environment that Carnegie provided to our young lab during that critical time. Today, I'd like to share with you our work on the structure, biogenesis, and engineering of the pyrenoid, a structure which is found in nearly every eukaryotic alga and plays a major role in the global carbon cycle. Pyrenoid is shown here in this beautiful electron micrograph from Robin Roth and Ursula Goodenough. Excuse me, sorry, I just want to get the pointer right. But before I tell you about the pyrenoid, I want to share with you why I'm so excited about photosynthetic organisms more generally. Photosynthetic organisms have this incredible ability to harness energy from sunlight and use this energy to assimilate CO2 into biomass. And the thing that I find most fascinating about them is the massive scale at which they do this chemistry. Globally, photosynthetic organisms fix CO2 at a rate of 400 billion tons per year, which is about 10 times the rate at which our entire civilization emits CO2 into the atmosphere. As a result of this massive scale at which they do chemistry, photosynthetic organisms impact nearly every aspect of our lives, ranging from regulating the global carbon cycle to making all the food that we eat, many of the fuels that we burn, and many of the materials that we use. Our world is facing major challenges in all of these areas in the coming decades, and it's the dream of my lab to contribute to addressing some of these challenges by advancing our basic ability to engineer enhanced photosynthetic organisms, such as crops that make more food with fewer resources. One of the biggest opportunities that we see lies in the enzyme Rubisco. Thought to be the most abundant enzyme on the planet, it performs this key reaction, fixing CO2 into biomass in essentially all photosynthetic organisms. But Rubisco runs slowly, and the slow rate of CO2 fixation limits the growth rates of many of our crops, including those of rice and wheat. Neither we nor nature have been able to engineer a better Rubisco, but it turns out that some organisms have evolved a way to make Rubisco run faster, about 10 times faster, by putting Rubisco into a compartment into which they pump CO2 to a higher concentration. Eukaryotic algae, prokaryotic cyanobacteria, and a few land plants called C4 plants have each evolved a different molecular way to achieve such a CO2 concentrating mechanism. And there's currently a great interest in understanding how they work and in engineering their components into the crops that don't have them. A lot of solid science suggests that if successful, such a transfer could increase yields by up to 60%, and the crops would require less water and less nitrogen fertilizer. Now, it's early days, and I think at this time, all of these CO2 concentrating mechanisms are worth studying. And my lab has focused on understanding the algal CO2 concentrating mechanism because it presents unique engineering opportunities and is the most poorly characterized. And we've done this as part of a wonderful collaboration called Combining Algal and Plant Photosynthesis, which started 10 years ago as a collaboration between my lab and the labs of two British scientists, Howard Griffiths and Alison Smith. And over the years, we've added many wonderful collaborators. So actually, Howard and Alison have recently retired, and the project on their end has been taken over by Alistair McCormick and Luke Mackinder. And we've also had really fruitful collaborations with Mark Stitt, Ursula Goodenough, Ned Wingreen, and Ben Engel. To help you understand what we're doing, I first want to explain the major difference between CO2 assimilation in plants and CO2 assimilation in algae. In both algae and plants, the CO2 assimilating enzyme Rubisco is found inside the photosynthetic organelle, the chloroplast. In a plant cell, the Rubisco is found throughout the chloroplast, and CO2 diffuses in slowly from the outside to be fixed by Rubisco. In contrast, in an algal cell, the Rubisco is clustered into a microcompartment called the pyrenoid, and CO2 is pumped from a low concentration outside the cell to a high concentration inside the pyrenoid, which makes the Rubisco run faster. Now, I want to show you what this looks like in an actual algal cell. This is a cross-section through a single cell of our model alga Chlamydomonas, which we call Chlamy. The pyrenoid is this little ball, which sits inside the chloroplast. It turns out that having a pyrenoid confers such a tremendous growth advantage that nearly all algae on the planet have one, including the algae that do much of the photosynthesis in the oceans: diatoms, coccolithophores, and chlorophytes. In fact, we calculate that about a third of global CO2 assimilation occurs in a pyrenoid. So this is clearly an organelle of major biogeochemical importance. And yet it's also one of the most poorly characterized organelles that we know of. If we zoom in on the pyrenoid, we can see that it has three subcompartments. There's a spherical matrix containing tightly packed Rubisco. The little clumps you see here are individual Rubisco holoenzymes. The matrix is traversed by a vasculature of membrane tubules, which we think deliver concentrated CO2 to the Rubisco in the matrix. And the matrix is surrounded by a thick wall, the starch sheath. When we started the lab 11 years ago, this was essentially the state of the field. The pyrenoid had remained almost completely uncharacterized at a molecular level. But if we dream of engineering a pyrenoid into plants, we first need to understand its basic molecular biology. What are its protein components, and how is it assembled? Which brings me to my outline. I've just told you that the algal pyrenoid enhances CO2 fixation. I'll tell you about how we developed new tools, including a mutant resource, to discover new components of the pyrenoid with high-throughput genetics. I'll tell you how we used a systematic localization strategy to understand the anatomy of the pyrenoid. I'll share with you our surprising discovery that the pyrenoid is actually a phase-separated liquid-like organelle and not a crystalline solid structure as it had long been thought to be. Then I'll talk about our molecular studies of the pyrenoid, which include the discovery of a key linker protein that holds the Rubisco together in the pyrenoid, and our discovery that Rubisco-binding motifs target proteins to the pyrenoid, and we think hold together the pyrenoid's three subcompartments. Finally, I'll briefly mention some early progress in our collaboration's efforts to engineer a pyrenoid into land plants. One of the biggest challenges that we faced when we started was that we knew almost none of the protein components of the pyrenoid. Having been trained in yeast genetics, we reasoned that we could make rapid progress on this question with yeast high-throughput genetics. All we'd have to do is screen the mutant library to identify new components and determine the localizations of hits to get clues for what each protein is doing. The only problem with these ideas is that yeast doesn't have a pyrenoid and it doesn't do photosynthesis. So we decided to develop these kinds of tools for an organism that does do photosynthesis and has a pyrenoid, the model eukaryotic alga Chlamydomonas. which is the leading single-celled model organism for the molecular biology studies of eukaryotic photosynthesis. It's also made major contributions to our understanding of cilia, interorganelle communication, lipid metabolism, and the channel rhodopsin widely used in optogenetics actually comes from Chlamy. Like yeast, Chlamy is a haploid which facilitates certain genetic studies. But when we started, progress in the hundreds of labs using this model organism was greatly limited by difficulties with obtaining mutants in genes of interest. There was no mutant library. The genome is 10 times larger than that of yeast. There was no effective nuclear gene targeting, and to this day, Cas9 is still fairly inefficient. Transforming DNA integrates at random sites in the genome, and mapping of these random insertion sites was mysteriously difficult. And finally, propagation was low throughput. Strains were propagated by hand in individual glass vials, as you can see here. To overcome these challenges, we started a massive team effort that was led by two outstanding postdocs, Xiaobo Li and Ru Zhang, who are now each running their own labs, with the help of an outstanding bioinformatician, Veronica Patena, who is still with us, and with the help of 6 technicians and a lot of advice from our collaborators, Pete Lefevre, who runs the Chlamydomonas Resource Center, and Arthur Grossman, who was our next door neighbor at the Carnegie Department of Plant Biology, where this project started. And together, we overcame the challenges that I mentioned and developed a genome-wide collection of 60,000 Chlamy mutants with known gene disruptions. This is a picture of several plates from our collection. In each of these colonies, a different gene is disrupted. 80% of the genes are represented. This is the first such resource in any single-celled photosynthetic organism. Over 5,000 of these mutants have been distributed to over 300 laboratories worldwide, and this resource is enabling advances in the broad range of fields that use Chlamy as a model system. The mutants can be searched on the website chlamylibrary.org, which also contains a large number of mutant phenotypes that we obtained by high-throughput genetics. Actually, we are just starting to put protein localizations and protein-protein interactions on this website as well. So I encourage you to consider using this resource for your work. One of the first things that my lab did with this resource was a massive screen to identify mutants with defective CO2 concentrating mechanisms. Leif Pallesen, a postdoc in my lab, grew each plate of mutants under 2 different conditions, air and high CO2. He then looked for mutants like this colony here in the middle of the zoom, which cannot grow photosynthetically in air, but can grow under high CO2. This classic phenotype suggests a defect in the CO2 concentrating mechanism. The screen yielded a list of candidates, and we next sought to determine the localizations of the candidate proteins. But at the time, it was very difficult to obtain the localization of even a single protein in algae. So Luke Mackinder, another postdoc in my lab, who now runs his own lab, developed new vectors and a pipeline that allowed him to tag over 100 candidate proteins from the screen and also from transcriptomics with the fluorescent protein Venus. And he then used microscopy to determine the localization of each protein. And we're excited to find that a number of these candidate proteins localized to the pyrenoid. This was intriguing because for the past 25 years, the pyrenoid had been thought to be primarily composed of 2 proteins, Rubisco and Rubisco activase, which is a chaperone for Rubisco. In these confocal microscopy images, each of the tagged proteins is shown in green, and the surrounding chloroplast is shown in magenta, visualized through chlorophyll autofluorescence. You can see that each of these proteins localize to a bright punctus in a hole in the chlorophyll autofluorescence, which corresponds to the pyrenoid matrix. So you can imagine that we were thrilled when we started to see other proteins that also localize to the pyrenoid, like this protein, which also localized to the pyrenoid matrix, and other proteins that showed previously undescribed localizations, like this protein that we think localizes to the starch plates, because when we take a confocal section through the middle of the pyrenoid, we see a ring. But when we take a confocal section through the surface of the pyrenoid, as you can see in this inset here, we see what appears to be starch plates with cracks in between them. Or this protein, which appears to form a mesh around the pyrenoid, and we hypothesize fills the gaps between the starch plates. Or my favorite, this protein, which forms a beautiful star-like pattern that traverses the pyrenoid, and we think corresponds to the pyrenoid tubules. We then went on to systematically do immunoprecipitations and mass spectrometry on these and other pyrenoid-localized proteins. And together, we identified a total of about 90 high-confidence pyrenoid proteins. The challenge now for the field is to use these pyrenoid-localized proteins as molecular handles to characterize the structure, biogenesis, and function of the pyrenoid's 3 compartments. In the next few slides, I want to share some insights these taglines have given us into the structure and biogenesis of one of the compartments, the pyrenoid matrix. The matrix had long been thought to be a crystalline solid. This view had emerged from electron micrographs taken 50 years ago, which showed crystalline arrays of particles inside the pyrenoid matrix of several species of algae, like this one. However, a crystalline solid packing of Rubisco leads to a paradox about how all of this Rubisco can be serviced by the much less abundant enzyme Rubisco activase, which is needed for efficient CO2 fixation. So Liz Freeman-Rosenzweig, a graduate student in the lab working with Luke, did a fluorescence recovery after photobleaching experiment to test whether the matrix is really a solid, and observed instead that the matrix mixes internally like a liquid. Here we're looking at a cell where the pyrenoid matrix is lit up by expressing fluorescently tagged Rubisco. These images are false colored, with red and white being the brightest intensities. In this experiment, we took a picture of the pyrenoid. Then using a laser, we bleached the fluorescence in half the pyrenoid, producing a pyrenoid that is unevenly fluorescent. When we watched the pyrenoids over time, we were surprised to see that the fluorescence homogenizes on the timescale of seconds, indicating that the pyrenoid matrix mixes like a liquid. These and other results led us to the conclusion that the pyrenoid matrix, at least in Chlammy, is not a crystalline solid, but rather behaves as a phase-separated liquid-like organelle like RNA granules and nucleoli. This new view of the pyrenoid matrix changes how we've been thinking about its biogenesis and engineering. How is this liquid-like state achieved at a molecular level? And what holds the Rubisco together in the pyrenoid? Luke found the answer in one of the candidates that he discovered, this matrix-localized protein, which Luke named EPIC1 for essential pyrenoid component. We now have extensive data indicating that this EPIC1 protein serves as a molecular glue that holds the Rubisco together in the pyrenoid. One of the results that supports this model is based on looking at fluorescently tagged Rubisco. In wild-type cells, fluorescently tagged Rubisco forms a bright punctus where the pyrenoid is. However, in a mutant lacking EPIC1, we see that the Rubisco is now found throughout the chloroplast, indicating that the EPIC1 protein is required for the normal localization of Rubisco to the pyrenoid matrix. Shan He, a research scholar in my lab, recently determined that EPIC1 and Rubisco each have multiple binding sites for the other, allowing the 2 proteins to form a dense condensate. In collaboration with HHMI's cryo-EM facility, Shan determined the structure of the binding interface, leading to our current model, where EPIC1 is an intrinsically disordered protein with 5 evenly spaced binding sites for Rubisco, which bind to specific sites on the Rubisco holoenzyme. Here I'm showing you 3 Rubisco holoenzymes that are held together by EPIC1 proteins. Now this is a snapshot in time, but in the real world, we think that EPIC1 and Rubisco bind and unbind rapidly, allowing the Rubiscos to flow past each other while staying together in a densely packed condensate. Our discovery and characterization of this EPIC1 linker protein solves the long-standing mystery of how Rubisco is held together in the pyrenoid matrix. And more broadly, our studies have now made the Chlammy pyrenoid matrix one of the structurally best understood phase-separated condensates in cell biology, making it a powerful system for studying the biophysics of biological phase separation. One of the big surprises came when we observed that these Rubisco-binding motifs are not present just on EPIC1, but are also present on dozens of other pyrenoid-localized proteins, like these, for example. In each of these proteins, we can see at least one Rubisco-binding domain, one Rubisco-binding motif that is depicted by one of these circles, in addition to other domains that we can see on these proteins, like these are starch-binding domains or these are transmembrane domains. Moritz Meyer, a research scholar in the lab, figured out that these motifs mediate the targeting of proteins to the pyrenoid. He established that the motif is necessary to target a protein to the pyrenoid by taking one of these proteins, which normally localizes to the pyrenoid matrix, when we fluorescently tag it with Venus. So here in green, you can see the protein tagged with Venus localizing to the pyrenoid matrix. And he showed that making a point mutation to this protein's Rubisco-binding motif now causes the protein to localize throughout the chloroplast, indicating that the motif is necessary to target this protein to the pyrenoid. In another experiment, he demonstrated that the motif is also sufficient to target a chloroplast protein to the pyrenoid. These findings have led us to our current model, where newly synthesized proteins that contain the Rubisco-binding motif diffuse around the chloroplast until they encounter the pyrenoid matrix, where they're captured by binding to Rubisco. So we think that this simple mechanism explains the pyrenoid targeting of many of the core pyrenoid-localized proteins. Now that we have a decent understanding of how the pyrenoid matrix is assembled and how proteins are targeted to it, another major outstanding question is, how is the matrix connected to the other two compartments, the traversing membrane tubules and the surrounding starch sheath? It turns out that the pyrenoid-localized proteins that I introduced to you earlier allow us to propose a model for how these three subcompartments are connected. On the left is our electron micrograph, and on the right, I'll draw a cartoon for what we think is happening. A lot of evidence suggests that the tubules can form independently of the matrix or the starch sheath. It turns out that two of the proteins that we discovered localized to the pyrenoid tubules. These are the proteins with the transmembrane domains and the Rubisco-binding motifs. We think that these two proteins link a layer of Rubisco molecules to the tubule membranes. Next, interactions of these Rubiscos with EPIC1, this linker protein, allow the pyrenoid matrix to coalesce around the pyrenoid tubules. Two of the other proteins that we discovered localize to the periphery of the pyrenoid matrix. And these are the proteins that contain the Rubisco-binding motifs as well as starch-binding domains. And this suggests to us that these proteins could link the starch onto the pyrenoid matrix. We're very excited about this model because it explains a growing number of experimental results. And to me, it seems like a fascinating example of how an organelle that at first appears to be quite complicated with its three different subcompartments and its complex geometry can actually be assembled based on fairly simple principles. And more broadly, we hope that this motif principle may provide insights into the organization of other phase-separated organelles across biology. We hope that our basic advances in our understanding of how the pyrenoid is assembled will pave the way for ultimately engineering this structure into land plants. And with that goal in mind, our collaborators, Nikki Atkinson and Alistair McCormick, have started to transfer the components that we're discovering in Chlammy into the model land plant Arabidopsis. And very excitingly, they've actually recently been able to reconstitute the Chlammy pyrenoid matrix in a land plant. First, I'm going to show you a wild-type Arabidopsis plant chloroplast. Here you can see the chloroplast with two big starch granules in it and the thylakoid membranes traversing it. In this wild-type chloroplast, the Rubisco is found throughout the chloroplast. Nikki and Alastair then expressed the EPIC1 linker protein along with a modified Rubisco, which can now bind to this EPIC1 linker protein. And super excitingly, they see that when they do this, the engineered chloroplasts now contain a single condensate of Rubisco, which they showed mixes internally, like we observed the matrix mixes in Chlamy. So we're super excited by this early success, and we look forward to working with them and with the rest of the field to hopefully complete the engineering of a functional pyrenoid into land plants. Now, it's important to note that these Rubisco condensates at this time don't do anything for the plant because they're not being fed by CO2. In order to be able to feed these engineered condensates with CO2, we now need to understand a second compartment of the pyrenoid tubules, which we think supply the pyrenoid matrix with concentrated CO2. Yet we know almost nothing about them at a molecular level. Here we're looking at a beautiful cryo-electron tomogram from our collaborator, Ben Engel. From these data, we can see that the tubules have several intriguing features. At the heart of the pyrenoid, they form a reticulated network out of which the tubules radiate. The tubules cross the starch sheath and fuse with the photosynthetic thylakoid membranes outside of the pyrenoid, which are actually sheets outside the pyrenoid. And if we take a slice through one of these tubules, we see that inside of it is a second set of membrane tubes, which are among the smallest known in any biological system, with an internal diameter of only 4 nanometers. How are these tubules made? How do they deliver CO2? Do they serve other functions beyond CO2 delivery? And how is the matrix connected to the tubules? It is my belief that answering these basic questions is going to make or break our ability to engineer a functional pyrenoid into crops. In summary, I've told you today that the algal pyrenoid enhances CO2 fixation. But when we started on this, we knew almost nothing about it at a molecular level. Because there were very few genetic tools available in algae at the time, we developed the world's first genome-wide collection of algal mutants with known gene disruptions, and used this collection to identify mutants with defects in the pyrenoid. We then used a systematic localization approach to identify even more components and to gain insights into the anatomy of the pyrenoid. I highlighted our discovery that the pyrenoid is a phase-separated liquid-like organelle and not a crystalline solid as it had long been thought to be. Then I told you about our molecular studies where we discovered a key linker protein that holds the Rubisco together in the pyrenoid matrix. And I shared our discovery that many pyrenoid proteins contain a common Rubisco-binding motif, which we showed mediates the targeting of many of these proteins to the pyrenoid matrix, and that we hypothesize actually mediates the binding interactions between the matrix and the pyrenoid tubules and between the matrix and the starch sheath. We hope that our efforts will ultimately allow the engineering of a pyrenoid into land plants to improve crop yields. And in fact, we're making progress on this with our collaborators. Finally, I'd like to thank the members of my lab who did the work that I presented to you today, both current and past lab members. Here you can see us with our Chlamy shirts, pretending to be Clammies. So Clammies have two eukaryotic flagella or cilia that they use to swim. And so we're pretending to have these eukaryotic flagella on our heads. We're also very grateful to our collaborators and to our funding sources. I also want to mention that we have a postdoc position available. So if you're excited about working with us on these problems, please reach out to me. Thank you for listening, and I look forward to your questions.